let's just jump right into the presentation. Today I'll be talking about the microbiology and immunology research I've done and give you an insider perspective towards the life in academia and the graduate school life through an international perspective, because I came here in 2011 for my Ph.D. So it was, maybe I was your age when I came here, and since then this is what I've done. So from 2006 to 2010, I was doing my undergraduate studies in India in B.Tech in biotechnology, and my dissertation title was Identification and Characterization of Clinical Isolates of Group A Streptococcus. That is a kind of bacterium which infects children and gives them strep throat. In older adults, it causes more complex infections, but I was focusing on group A Streptococcus, which is more for the children. Then in 2011, I took a very different route. Usually, a PhD student does his master's and then decides on the PhD, or gets the master's training, does some courses, probably do a little bit of research, and then jump into the PhD. But I think I was too much in a hurry, so I directly applied for PhD courses here in USA, and I got a few acceptance letters, and I found the University of Florida's Microbiology and Cell Science Department's PhD program the most appealing to me, and that's why I joined it. And over there, my research topic was characterization of sec-translocon dependent extracytoplasmic proteins of Candidus Libibacter asiaticus, which is the causative agent of citrus greening, as you all must be knowing. We'll be talking more into detail about my PhD research and the research which I'm currently doing after graduating from my PhD in the same department, because I thought that it was very rewarding working with a plant pathogenic bacteria, but I also wanted to do something which was close at home for me. And Leishmania is an infection which does affect the Indian subcontinent as well, amongst the other parts of the world. So I wanted to go more into something which is more close at home. And that's why I'm working as a postdoctoral researcher for uncovering the molecular composition of the parasitophorous vacuoles in which the Leishmania parasite resides within the host cell, and the identification of novel targets for control strategies. So a very good question when I was practicing for this talk, myself asked that probably people don't know what a postdoctoral researcher is. So just to tell you more about it, it's more of an in-between position. Once you graduate from your PhD and before you take up a job as a faculty position in academia, it is very important, because as a PhD student you're working on one project and you're only working on that project and you're a student. So it's very important to know how to manage a lab, to mentor other students who are graduate level or master students. So that's what the job of a postdoctoral researcher is. That is also a kind of training which is an preparation training to be a faculty position. It is also a good training for being at a managerial position for a company's scientific industrial unit. So I'll be talking about these two researches more in detail. So just to give you an outline, first we'll talk about the research work which I did in my PhD and my postdoctoral, currently I'm doing in my postdoctoral studies. And then I'll be going for a more personal perspective about the reasons why I opted for a PhD, the international experience I had during my graduate school life, and more in detail about the graduate school life, and then of course the future in the field of academia, how that correlates with the PhD degree which you sought to earn. So my first topic is regarding citrus greening, which you all must be very familiar about here in Florida. So the citrus industry is actually very, very economically significant for a state like Florida, where citrus is one of the preliminary crops which are grown. It has a global in Florida itself, it has an impact of about $9 billion annually, and it also provides thousands of jobs in Florida, and also creates a lot of revenue through trade and export. So we all love citrus. It's very nutritious. It boosts our immunity, and who doesn't like their OJ in the morning? So that was all the good news. But the bad news is citrus greening, which is also sometimes abbreviated as HLB. So in 2005, we first identified citrus greening in Florida, and annually it has started costing us about $3.63 billion in losses, and about more than 6,500 jobs have been, people have lost their jobs because of this. And this is just the 2010 consensus. So the numbers are much higher now in 2016. It has been more than 10 years now, a decade of citrus greening affecting Florida counties. And this map is only till 2007, but till now the whole of Florida has been affected by citrus greening. So it is definitely a very devastating disease for the citrus industry, and especially for areas which are mainly focusing on the crops such of citrus plants. So HLB, or citrus greening, is actually caused by the Candidus liberbacter species, and mainly in the Candidus liberbacter species, the genus Candidus liberbacter asiaticus, africanus, and americanus, based on where they were first isolated from. So the Candidus liberbacter asiaticus species is, or LAS as we call it, is the most prevalent one in Florida, and that was my focus of research. So a little more about the Candidus liberbacter asiaticus. It is a gram-negative alpha proteobacteria which belongs to the Rhizobaceae family. The term candidatus in its name signifies that it cannot be maintained in lab culture as yet. So that makes it even more difficult to work with. Something which you cannot grow in lab, it is difficult to study and do experiments on it. So all these studies which have happened are either field trials or field studies or greenhouse studies, or they are bioinformatics studies which you do on the computer based on your previous knowledge regarding very closely related species. So this, even though it makes it more difficult to study, it definitely makes it even more fascinating, because you want to see why it's not growing like other bacteria do. So it has a very different kind of life cycle, which I will be talking about very soon. But it has two hosts. One is the plant host, which actually gets affected by the citrus greening infection. But it also has a vector host, which helps it to move from one tree and infect the other fresh tree. So the vector host is the Diaphragma citri, or the Asian citrus psyllid, and when it infected psyllid bites on the plant, it introduces the bacterium. Here you can see there are many psyllids feeding on the leaf. It introduces the bacterium into the plant's phloem tissue, and you can see that there is blockage of the phloem tissue due to the deposition of or the spread of the bacterium inside the tissue. So basically the phloem tissue of a plant is like your veins or arteries, which provide nutrients to other parts of your body, and similarly the nutrition and transport of nutrients happens through these tissues. But when they get blocked because of this infection, the plant starts losing nutritional value in different certain areas of the plant, and ultimately leads to the decline of the tree health, and the tree after an HLB infection usually dies within 4 to 5 years. So there are growths, full growths which have got infected with citrus greening and are now currently not putting any fruits. So the citrus greening infection cycle is very fascinating because the hosts are so different. The one is an insect host which basically acts as a vector or a carrier from an infected tree to a fresh tree, and the other is the plant host which has a completely different physiology, and the bacterium resides within the cells of the plant tissue. So a very important question is that based on this life cycle, what proteins are helping the bacterium to survive in such different kind of environments, and also not to be able to be recognized by the host's immunity. So plants also have some immunity, the insect have its own immunity, but they're not, but this bacterium manages to completely hide from the immunity. So for that, my project was more focused on sec translocation mechanism in LAS. It is a kind of protein transportation pathway which is elucidated here, but not to go so much into the science of it. Basically, if this is the extracellular space and this is the cell cytoplasm, the proteins are being created inside the cell of the inside the bacterium cell, and this bacterium cell transports these certain specific proteins through the sec translocation pathway. This is the pathway which consists of many different proteins. So it transports the proteins which are formed, not all of them, but very specific ones, transports these proteins and secretes them out into the extracellular space, which provide it environment to be able to sustain itself in the host, inside the host cell. So I wanted to know more about the sec translocation mechanism in LAS, Candidus Libibacter asiaticus, and characterize and identify these secreted proteins of LAS. So like I said, that these proteins might be playing important roles in the pathogenicity of the bacterium, and also helping it to establish the infection and sustain itself inside the host cell. So it wasn't as easy as just to go and take a look at it. We had to do many bioinformatic analysis, and then because bioinformatic analysis is all online on a computer based on very closely related other proteins, or based on very closely related other species. So it was important to validify them experimentally as well. So what I got results was that sec translocation is a major route of translocation of proteins in LAS, because it's the only one. Usually a bacterium has about 7 different types, not all of them, but usually they have 5 to 7 different types of translocation pathways through which proteins are transported outside of the cell. But in LAS case, because the genome size of LAS is so small, only sec translocation pathway is the major pathway through which proteins are being specifically secreted out of the cell, or at least into the periplasm. So another thing, bioinformatically, I predicted putative 150 candidate proteins of LAS which might be getting secreted and ultimately helping the bacterium to sustain itself and establish infection. So I wanted to experimentally confirm these extracytoplasmic or secreted proteins, and based on my experimental analysis out of the 4A assay, which is a very dependable biochemical assay which you perform on proteins, I confirmed 89 proteins of LAS which were being secreted outside of the cell through the sec translocation pathway. Why I say confirmed 89 and I do not just say only 89 is because the bioassay could confirm 89. This does not mean that the rest of the proteins are not secreted. It's just that they cannot be confirmed because we don't have the organism growing inside the in our lab conditions. It's difficult to do biochemical assays. So that's why there was this limitation with the study. But anyway, this study is being is actually in review now and will be published in a peer-reviewed journal. So hopefully that should be this month. So that was my PhD research. Now coming to my postdoctoral research, which I'm currently doing, I wanted to do something more in the same area of study, but I wanted to do it in an animal system now, because before that I was doing it working with a plant pathogen. So in my department itself, there's a lot of very good research going on, and I happen to have the opportunity to work on characterizing secreted and associated proteins of Leishmaniasis causing parasite, which is Leishmania species, and that's my postdoctoral research which I'm doing currently. Don't mind the pictures, they're a little graphic, but the Leishmaniasis infection is actually a very devastating infection, especially in the developing countries in the world. It is a parasitic disease caused by the infection with Leishmania parasites, and it is spread by the bite of infected sandflies. So you can see that there is parallelism between my PhD research and my postdoctoral research. Both are both kinds of one bacterial pathogen and one parasitic pathogen cause diseases in animal cells, but they are being vectored by an insect host. Also, both are intracellular. So the in case of citrus greening, the bacteria resides within the plant cell, and in case of Leishmania infection, the Leishmania parasite resides within the a type of immune cells. You must have heard of them, macrophages, which are the first line of defense sometimes. So that's where they reside. So there are many different forms of Leishmaniasis you can have based on the species which has affected you, and also based on the country that species is from. So the different forms of Leishmaniasis are cutaneous Leishmaniasis, which you can see here, which basically is more easier to identify because you develop skin sores and ulcers, and they are painful sometimes they're not painful, but they can be easily treated with miltefosine treatment. And unfortunately, in some developing countries, these are not, because of the not lack but scarcity of medical facilities, this can be ignored and then it may turn lethal. And then there is also visceral kind of Leishmaniasis, which is more severe and can be lethal if not treated at the correct time. And in visceral Leishmaniasis, you have internal organs which get affected by the parasite. So in this particular picture, the spleen is getting infected, and it gets enlarged because it's so full of parasite, and the immune system is trying to defend it, but at the same time the infection has established a lot, progressed a lot more. So internal organs consist of spleen, liver tissue, bone marrow sometimes, which is very difficult to treat, but if given proper treatment and care, visceral Leishmaniasis can also be completely eradicated from your body. Another thing is that some people do not have any symptoms, especially for visceral Leishmaniasis. That can be very lethal, and you actually recognize when you have the time point at which you recognize when you have got a Leishmaniasis infection is when you acquire a different or co-disease, because your immunity is already compromised because of the Leishmaniasis infection, and then you acquire another disease which is just lurking for an opportunity to come by. So as just to give you a picture of the impact of this infection, the cutaneous form, there are about 1.2 million cases annually and globally, and visceral, it's about 0.4 million cases. So it is very prevalent all over the world. It's not present in USA and many cold climates, but most of the tropical countries have it, except for Australia, because probably it's not reached there, the sandflies. Anyway, but it's present in more than 90 countries, and like I said, that it's also affecting the Indian subcontinent, so I have a more closer motive to work hard on this. So this is what I'm doing right now. The Leishmania infection cycle is very interesting, and it is very parallel to the citrus greening life cycle, even though we cannot compare both of them. Citrus greening is a bacterial disease. This is a parasite, which is a eukaryotic organism. But still, just to compare, the infected sandfly takes a blood meal of a healthy individual, and the promastigote form of the parasite are get entry into the healthy individual. These promastigote forms, once inside animal cell, transform into amastigote forms, which are not motile. The promastigote forms are motile because they have to go and reach the cells. The amastigote forms, once they are inside the cell, they don't need to be motile, they need to be multiplying faster. So the amastigote forms multiply really quickly. Like it said, like this is the diagnostic stage, and this stage, if you catch it, it's very good. You can do the treatment and you'll be fine. So once they multiply, they infect either other macrophage cells, or if a sandfly bites an infected individual at this stage, this sandfly starts carrying the infection, and then the amastigotes convert into promastigotes, and then the cycle continues like that. So just to give an overview about the Leishmania parasite, like I was saying, the flagellum is highly developed in the promastigote form because it needs to be motile to find the particular cell it needs to be dividing inside of. Once it's inside of the cell, like you will see here, these are Leishmania parasites, which are, this is one cell, this is the nucleus of the cell. So it's, once they are inside the cell, they convert into amastigote form, where they don't need to have the flagellum, but they do need to have all the other metabolic organelles to be more active. So this is from Leishmania mexicana species. So they have this kind of vacuoles which form. This picture represents the Leishmania donovani species, and this is a macrophage cell. And you can see that this cell has been affected by one parasite, Leishmania donovani parasite. And you will see that the parasites which are outside, they are still in their promastigote form. So it's very interesting, and it's very interesting to view it every day under the microscope and just see and take a look at their behavior, even though all that has been documented already, but it's just fun to just view it and see how it works. So the project objectives, this is another, the N represents the nucleus and the V represents the vacuoles, which contains the parasites in it. So the project objectives in this project is that the parasites reside in the parasitophorous vacuoles, which may play an important role in the parasite survival inside the cell. The LPVs are conduits of parasite strategies. These strategies are basically proteins which modulate the host cell activities in their favor. It's like a virus on your computer. So the virus starts doing stuff for your computer to make it work according to its own programming, something like that. And then understanding the molecular composition and characteristics of LPVs, which will help us in determining their role in Leishmania pathogenesis, and then this knowledge will ultimately help us in identification of novel targets to develop a new strategy of control, which basically means that if these parasites are releasing proteins inside the cell to help reproduce, if we target those proteins which are secreted with a drug, then those proteins will not be able to help the parasite to reproduce any longer, and then it can be treated very quickly. So it's very important to identify those proteins which are helping it.
I have a question.
Yes.
So those vacuoles,
Uh-huh.
how did the parasite get in? So did the cell eat it like endocytosis,
Yes.
the lysosome?
So the macrophage cells, these cells are phagocytic cells. They engulf any foreign particle, or in this case parasite, or in fact bacterium cells, or any foreign particle which is non-self to the body. They engulf it, and either they lyse it, or if it's not harmful, then they just excrete it out. So it's very actually easy for the parasite to get engulfed by the bacterium, but what's interesting is that the macrophage cell is not able to lyse it, but actually the parasite is forming a vacuole within it and actually making a home out of it. So that's very fascinating. So all this is about research work. Coming to a more personal experience throughout my graduate school life, I would come directly why I joined the Ph.D. program. So I always wanted to be in the field of academia. So that was definitely one of the main reasons why I was looking into going into a Ph.D. program. But definitely there are many other things which go into deciding on a Ph.D. So the main and most important thing to at least me was that you would achieve something significant. You would have gone through a whole experience to obtain not just a degree, but also obtain a knowledge and understanding in a way which you usually do not do in your general curriculum. And of course, it's a long procedure. It's a very big commitment. So that's why the meme over here. You also learn or discover something new. It's not something which has ever been done before, even though it is very narrow, and it's not like you are, you're not being Columbus and finding a whole new civilization, but what you're doing is very narrow and very specific, but it is novel. It's your work, and you have found those results. Nobody in the world has ever found out those results till then. And those results will be used to find out more stuff later on by other researchers. So that was very rewarding to me. It's also the drive for research. I've always been a very curious child, and people can take out their curiosities in many ways, in creative ways, in sports, in many different forms. And for me, it was research. You got to play with biomolecules and organisms which you can't even see. And it was just fun because, at least to me, it was very interesting and fun because you're doing something which you used to read in textbooks, and now you actually see them. I remember the first time I extracted DNA out of plant tissue, and because it was so much DNA, so it precipitated in the test tube, and I could see it first time, all that DNA from precipitated DNA through my naked eye. So I got so excited. I wrote in my journal that this is the first day I saw DNA through my naked eye. And so you have to find what interests you and what drives your curiosity. Another thing is that you do become an expert in the particular area you are working at. So about my PhD, of course, my professor does know a lot about in that area, a lot of knowledge in that area, but my dissertation thesis PhD is something I would be knowing the most about. So you are the expert. Even in that little area of 90 pages you have written, you are the expert. And that's why actually celebrities get the PhD, because they have achieved a level of expertise in that particular field. You also get to enjoy the academic environment. I think the academic environment is really fun. A lot of people make fun of that, oh, you're a professional student, but it's actually fun because you get to be in the on the campus, you get to see many students coming, you mentor so many undergraduate students, you take up many new classes, anything new which has come in your area of knowledge, you can take a class about it, you can interact with different groups and researchers, you can interact with professors. It's actually a very good environment to hone your skills in. You also develop important transferable skills. So what was very important to me was that I wanted to go and teach further in the field of biology. And in order to do that, you don't need to only have a degree. You also need to have skills to be able to teach people, to be patient enough to go with every kind of student. And those are the kind of skill sets which I also learned during the PhD. It's not just the scientific skill set of bioinformatics, of microbiology, of biochemistry, but it was also the life skills which you learn over the process of PhD. So those are very important. And of course, you get paid to study, so that's very good. Of course, this picture is probably an overreach. You do not get paid a lot, but that is enough to not let you worry about paying bills. You're not working to pay bills then. You're working and your bills are getting paid by the stipend, and what you're doing and focusing on is your actual work. So you don't have to do 2 jobs. And I mean, it depends from case to case, but it's always fun to earn something for studying. It's quite an incentive and push for you to work more. And of course, at the end of it all, you achieve a prestigious title, and that was very important to me. You're a doctorate, everybody respects you. And it's not just about respect, it's about the fact that you don't even have to prove it, but everybody already can see that you committed to something and you achieved it. So that can be done for any field, but in field of academia, PhD is probably the easiest way to commit to something and achieve it and prove your worth, basically. So I think those were my reasons. There are variable reasons for which people do. Some people do it because they didn't get into med school, but there are different reasons. Whatever your reasons are, there are definitely many benefits out of it. So coming to the international perspective, I don't think you will be able to read this now, but it's just the F1 student visa process in a very dark humor kind of way. But I was from India and a bachelor student. I was 23 when I came here for my PhD. And frankly speaking, it was a lot of difference. It was not more difference. I'm from New Delhi, India, which is already a very, very big city. So coming to Gainesville was actually coming to a smaller city from a very big city. But in terms of college life, it was way different. Firstly, the credit system. So it was very different for me. In India, you usually have a percentage. So you give an exam out of 100, you get whatever percentage you get, and that's your score. But credit system is much different. It's more adaptive to the particular set of students. So that is something which I learned here, and I adapted very quickly to it. So I would advise you that if you do get an opportunity to work or study abroad, try to get the system very quickly adapted to your lifestyle, because it will be beneficial to you. I could transfer a lot of my undergrad credits to my graduate school because of getting to know that, okay, my percentages which are really high over there, I don't have to take another class of the same thing, and I can actually just transfer it, and I'm actually getting an A in it now. So that's really cool. Even though on percentage system, if I got 81, through credit system, you can actually try to get an A if the whole class is getting a curve. So all those systems, just to tell you that it's very important to know the system you're working with and try to make it work according to you. That's what I did. The college life is definitely different. College life is more independent, I would say. You choose your classes, you choose according to which semester, according to which point of time in your life you want to take which class. If you're already taking a busy or difficult class, you don't want to take another one. So that kind of independence was very, very, I would say, very fortunate for you guys to have, because in India you usually have a curriculum set. So in each semester you have a curriculum of classes which you will have to take, and that's the semester you take it. Whatever you score on it, that's your grade. So you better be good about it. So that's something which I think is very student oriented. The college life, other than that, I mean, I did not come for an undergraduate school, which probably would give me more in-depth knowledge about sororities, frat houses, this and that. But other than that, the college life was very nice. The students were from all over the world, especially because USA, people from all over the world come. So that was very nice. I met people from many different countries, experienced many different cultures. And the college life is very student oriented, and you have different groups for the kind of activities you like, the hobbies you like. So that was very nice. Another very important thing which I found out here is that you're not spoon fed. In India, I would say that the culture of going to a college is almost mandatory. If your parents or you can afford it, you go to college. So over here, it's not like that. It's not necessary for you to go to college. It's your will to go to college. If you want to go to college, you go to college. And that's why there is no spoon feeding. Nobody is pushing you to do something. It's up to your curiosity and your commitment and your drive to go and do or get an A grade or get a B grade. It's up to you, and the professor can give you and guide you actually, but they're not going to spoon feed you with each and every answer. And that's something which I really like, because that kind of makes you help you decide whether you really want this or not. If you really want it, that's only when you will work for it yourself with your own motivation and not because somebody is pushing you into it. So I think that's very, very good about student life here. Another thing which is very specific to, I would say, PhD is that there are phases when you are treated as a student, and then there are phases where you're treated as a professional during your PhD. And I think the balance between the two is very important. And the balance, how to get to know when to act in which role, is very important. As a PhD student, you might have undergraduate students who will be volunteering in your lab. So for them, you need to be professional. You need to mentor them as a professional. But to your PhD guide, you're always going to be a student, and it's best that you're a student because then you can imbibe as much knowledge from them. So I think that balance is something which I initially struggled with, because I was 23, and then sometimes I had to act like a professional, but at the same time, sometimes I had to be a student. So it's a little, you have to wrap your brain around it, but once you get the hang of it, it's probably the best. You can be a student when you want to watch a free movie being showed for the students in the college, or you can be a professional and get all the benefits of healthcare and dental insurance. So all that is very nice. A very, very important factor was the teaching experience, because initially from day one, my plan was to be in the academia higher education system itself. So the teaching opportunity, or the TAing, which I did, was very, very beneficial and crucial to me. The TA it was a TA position, but we were given our own. They were like, you are PhD students, you can handle 2 sections. And here are those 2 sections, you're in charge. So it was very interesting. It was a lab class, microbiology. And it was very interesting to see students from different courses, pre-meds, nursing, and many other courses who would, or even microbiology majors, to come and learn. And you are nervous yourself because you were an undergrad a year ago, so now you're teaching those undergrads. So but it's very rewarding. I think if you can teach somebody, you're probably helping them in a much, not helping, but probably giving them a very good gift. And I know it sounds cliched, but I can say that with experience. And I think any teacher would say that with experience because it's very rewarding to see a student get excited about this field of study you are excited about, and you have created that excitement in them. So that's very rewarding. And of course, it was a new culture for me. India is very different from USA in terms of culture. Some things which are very, very taboo in India are very okay here. Some things which are very okay in India are very taboo here. So it was very interesting to know that. Although I'm from New Delhi, so it's a very big city, and there's there's a lot of Western culture influence there. So it wasn't a shock, I would say, cultural shock, but it was definitely a very, I would say, learning experience, and it was fun. I think now I can go to any country, at least English-speaking countries, and be very easily able to interact with the local people there, learn about their stuff, because coming here, going through the same education system as they are going, and just learning about them gives you a very good, not just boost of confidence, but also a boost of curiosity. So the graduate school life, I can break it down to the kind of stages you go through. You get accepted, then you come here, and then you do rotations in different laboratories, and based on that, you choose a lab. It's not just that you only choose a lab, the lab also has to choose you. The PI has to, the professor has to like you and your work, your way of working, your lifestyle, that if the lab works from 8 a.m. to 6 p.m. and you are used to coming at noon, then of course that lab is not going to choose you. So it's like almost a mini marriage, and if it fits well, then it's really good. So you choose a lab, then you choose a committee. A PhD committee consists of 4 or 5, sometimes 6 professors, which are basically your PhD committee. What they do is guide you throughout your course, since after choosing all this is guided by the committee. So when you're probably in your first year or end of first year or second year beginning, you do your first committee meeting where you explain what your project is. Of course, by that time you have no results, and even if you try, you have no results. So you basically just introduce your subject to them and your project to them. Based on that, they give their inputs, and based on that, you try to do experiments and plan your experiments. That's very important. Then you have written qualifying exams, so this qualifying exam is basically after 2 years into your PhD as a PhD student. What this qualifying exam is that it makes you a PhD candidate. Before that, you were a student. After qualifying for your PhD candidacy, you are a candidate. That means that you do have the aptitude to be a researcher, to think scientifically and plan everything scientifically. But before that, you're just a student. You're learning how to be one. But if you don't have the aptitude for it, then there's really no point doing it. So you have written qualifiers, which is more subjective based on your committee. The oral qualifiers is more about your research, what you've done. So you present what you've been doing, and if that sits well with the committee, if they think that is scientifically relevant and sound, then they pass you and you are a PhD candidate, which is really a milestone. Then usually you have more committee meetings depending on how long you're taking. I did this in 4 years, 4.5 years. But some people take longer to finish their PhD, so then they have more number of committee meetings. Then, of course, in your last semester, probably you're writing your theses and manuscripts about your work. That itself is a very long procedure. You write, then you send it to your professor, then they make corrections on it, then you make those corrections, then you send it again, and then they find new corrections and probably some old ones, and then it's a process. But it's actually, now that I think about it, it's actually fun in itself, because you're like, you get up in the morning, you have your coffee and you're writing, and then you get out probably in the evening, and then it's nice to look back at it, because you feel that, okay, I have that capability. I never thought as an undergrad that, oh, I don't think I can do that. But now that I think of it, I'm really happy that this PhD experience brought that out of me. So, and I think it can bring it out for anybody, because you want to write your thesis. Then your thesis finally gets approved, which is the most important part. And then you defend your thesis. So basically, you give an oral presentation about your thesis and your work, and then people ask you questions, and you defend those questions. They ask you scientific questions, and then you defend it scientifically, proving why you did that and why it's scientifically sound. And then you graduate. So PhD life, graduate school life was like a family to me. This is our lab with their family members. You meet people from around the world. One day we had international night, in which we had food from every kind of country, and we put flags on it so to identify whose food it is. You're always in your lab. This is your second home, or probably your first home. You enjoy the lab life, though. It's really fun. You go to very new and exciting places and present your work. My poster is cut out here, but you present your work either as poster or oral presentations. You get to teach, which is always fun. At least for me, it was a lot of fun. And then you graduate, and you're really happy that it's finally done. And the future prospects after PhD are, it's actually up to you. You can do a PhD and then go into acting. That's not a problem. But usually what people take the route of is that they become postdoc for academic jobs, and which is basically a pre-faculty position. It's research oriented. It's more focusing on the management of a lab and how to run a lab. The salary is a little lower, but I would say that you're learning too much. The industry job and startups are what people do for more lucrative options. It's more applied research in the industry job, and the startups, whatever skills you've learned, you try to market them. And then some people go for further studies, MBA, MD, and probably work for the many different directions after that, government policy making jobs, who will get which grants to get approved, which grants not to get approved. And also sometimes we see on in like very popular shows, some PhD people get to work with NASA, FBI, CDC, and all those really fancy jobs. So, and work with zombie-related viruses, so it's fun. So with that, I would like to acknowledge everybody throughout my PhD experience, especially University of Florida, which accepted me and gave me the education. My PhD lab and its members, my professor, Dr. Nian Wong, the funding agency, which was for my PhD research, my current lab, which with Dr. Kima and my lab mates. I don't have everybody's picture there because I couldn't find it. And we're yet to take a lab picture with me in it. So the funding agency for my current research about Leishmania. And a very big thank you to all of you for going through and giving your attention. And with that, I will take any questions if you have.
Thank you.